Our next presentation is entitled to Seed Coat Biochemicals Mediate Aspergillus flavus Resistance in Peanut, and our presenter is Leslie Commey.
Good morning, everyone. I'm Leslie Commey, speaking from Texas Tech University, a master's student. Today, I'll be talking on seed coat biochemicals mediating A.flavus resistance in peanut. Peanut, we all know, is an important leguminous crop which is grown in the tropical and subtropical regions of the world. And it is used as certain food components such as oil, such as peanut butter, and also boiled peanut, which is famous in Atlanta, Georgia. And also it's used as a protein source for animal feeds. And also it's also used as a cover crop in certain agricultural research. However, 2/3 of its production area is found in developing countries, which is characterized by drought and heat stress conditions. Drought and heat stress conditions are the major requirement for A.flavus infestation to take place in the field. However, A.flavus and A. parasiticus are known to cause aflatoxin contamination, which is one of the notable diseases found in peanuts, with A.flavus causing aflatoxin B1 and B2, and A. parasiticus causing aflatoxin G1 and G2. However, A.flavus contamination also occurs during pre-harvest and post-harvest condition. And during that stage, the seed is about to enter into dormancy, so there is no active genetic resistance present at that time. And aflatoxin contamination has resulted in many peanuts from these developing countries being rejected by the EU since they are not able to meet the 4 parts per billion, and they don't have any techniques that can be able to determine whether their samples are contaminated or not. Some of the techniques that are already in existence are ELISA tests, which is very expensive, and the farmers are not friendly with using it. However, in an attempt to solve this problem, certain host pathogen interaction parameters have been developed, such as in vitro seed colonization and aflatoxin contamination percentage, and also pre-harvest aflatoxin contamination to identify resistance lines and see whether they can introduce it into breeding programs. It's still ongoing, and we hope to see something. However, coming up with a quick and less expensive method to quantify aflatoxin contamination is the way to go, in the sense that since it's 2/3 of its production is coming from developing countries, if you are able to find a farmer-friendly technique and also we are able to identify A.flavus at a faster pace, we can be able to know whether these samples needs to be exported or not. And A.flavus being a soil pathogen, a seed pathogen, the seed coat after processing is what is the only layer protecting the peanuts, so could provide an understanding to know whether or could be exploited to find out if we have resistance or its attributes or contributes to resistance to A.flavus. This led us to do a preliminary study using one of the host pathogen mechanisms known as in vitro seed colonization, which have been used to identify resistance lines. So based on that, we did an in vitro seed colonization for 55437 and TMV2, and we placed them under a temperature of 28 degrees for 8 days. And based on that, we found out that 55437 showed greater resistance as compared to TMV2, and the fungal DNA for 55437 was lower as compared to TMV2. This led us to hypothesize that seed coat could contain some biochemicals which will mediate A.flavus resistance and could be exploited to find a quick and efficient method to reduce aflatoxin contamination. So our objective was to see whether the seed coat biochemicals played a role in A.flavus resistance using radial growth bioassay, and also identify other resistance lines based on their seed coat color via in vitro seed colonization. So first of all, what we did was we took an old peanut stock, put it on a suitable media, PDA media, And then we were able to isolate the A.flavus strain. So this picture depicts both A.flavus and Aspergillus niger. So we took the A.flavus alone and we subcultured to obtain the pure strain of A.flavus. After that, we wanted to confirm, because the green color alone could not only give us an idea whether we've obtained a pure strain of A.flavus or there was a contamination. So what we did was we used molecular characterization, basically PCR method, and we found out that, so with the L2, the A.flavus gene is supposed to have a band size of a gene size of about 538 base pair. So we found out that within our 5 samples that we placed, 4 of them were able to contain the A.flavus. Then what we did further was to see whether the A.flavus strain we've obtained was aflatoxin, was containing aflatoxin or not. So we used the aflatoxin gene precursors such as AFLTIS and AFL0 to see whether our strains contains aflatoxin. So what we realized was 3 of our strains contain both, and we also use other methods to see whether we can be able to determine whether it's aflatoxin or not. So we also use the chemical method where we use beta-cyclodextrin and ammonium hydroxide. So with beta-cyclodextrin, what we did was we used 0.1% of beta-cyclodextrin and viewed it under UV. And if aflatoxin is known to be a fluorescence nature, so if it's fluorescent, it shows that there's presence of what? Aflatoxin in the strain I'm holding. Then also we expose this to ammonium hydroxide vapor. And this, the first one on my intermediate within my picture, shows a red bulb color, which indicates that there is aflatoxin in my sample. And the control, there was nothing like red bulb color. So we boldly said that, okay, the strain we had was A.flavus and it contains aflatoxin. This led us to our first objective, which what we did was we crashed the seed coat alone from 55437 and TMV2 and used acetone, acetic acid and water in a ratio to obtain or extract the biochemicals from it. And we introduce it into a PDA media. Then 1 times 10 raised to the power 7 of the A.flavus strain, which was quantified using hemocytometer, was introduced into the middle of the PDA media, and daily data was taken till the control, which is a full Petri dish with none of the 50 biochemicals from 55437 and TMV2, just water, was full. So on the 8th day, we realized that the plate was full, and the average data we took, we repeated the experiment for 3 times, and based on that, we analyzed the data for statistical for significant difference using Hanover. What we realized was in our 3 experiments, we saw significant difference between TMV2 and 55437 as well as the control. Based on this, we requested different seed coat colors from USDA to see whether we can use only just the seed coat phenotypic color to ascertain whether we are obtaining an A.flavus resistance line or not via in vitro seed colonization. What we realized was among all our lines, PI 544346 showed a greater resistance than even the known resistance, which was 55437. This was a very interesting result. So what we did was we looked at comparing the samples, the 11 samples we obtained. We realized that although PI 561680 and PI 561674 were a little bit below the known resistance, we took this and the PI 54346, and we are going to currently grow them under drought conditions and also use it in our next steps. So our future step is to be able to use HPLC analysis, which is a wet chemistry method, to identify the specific phenolic composition and also know the quantity they are in the seed coat and also use radial growth analysis to exploit and see whether those specific phenolic compounds will be able to mediate or regulate A.flavus growth. And also, based on the specific phenolics data we have obtained, we are going to develop a dry chemistry method, which is going to be a farmer-friendly technique so that it can be used in developing countries, and also breeders can incorporate into their breeding programs to develop a resistance, to know or ascertain whether the samples they have after crossing are A.flavus resistant or susceptible. Also, the resistance line we've already obtained from USDA will be grown, those which showed resistance are going to be grown under drought condition, and will evaluate for pre-harvest A.flavus contamination, and also look at the specific phenolic composition, whether drought conditions has any role in increasing maybe the specific phenolic compositions. Also, what we want to also do was to produce an A.flavus resistance line from cross between TMV2 and 55437. Currently, we are on the F3 stage on the field. However, because of the COVID, we weren't able to ascertain whether it was a true hybrid or not. But we hope that after we are going to take some leaf samples on the field and see whether we can see whether the crosses we did, we obtained heterosis or heterosis lines. And also, we also plan to use the new resistance line, which is PR544346, cross it with TMV2, hoping that by the time it will reach about the F3 stages, our biochemical or our specific phenolic and dry chemistry method has been obtained, and we'll use in conjunction with in vitro seed colonization to ascertain whether it's resistant or susceptible. So from this, we want to say that seed coat biochemicals will contain certain compounds which could regulate A.flavus growth and could provide an efficient tool or an effective tool to screen aflatoxin contamination, and also could also provide an alternative to be used with the other three known resistance identification mechanisms such as in vitro seed colonization, pre-harvest aflatoxin contamination, and aflatoxin contamination percentage in peanuts. I want to further acknowledge USDA under the Peanut Innovation Lab for sponsoring my research, and also USDA Grain for providing us with the seed samples, and our partners in IVSC. Thank you very much. Any questions, please?
Thank you very much for that presentation. And we do have a couple of questions from the audience already. The first question comes from Jianping Wang. And the question is, how did the seed coat biochemicals were retrieved and quantified before they were added to the medium? So I think they said, how were the seed coat biochemicals retrieved and quantified before being added to the medium?
So the question is, how was the seed coat quantified and retrieved before it was added to the PDA medium? So what we did was we crashed the seed coat. So it was a one generation late peanut lines that we obtained. That's the 55437 and TMV2. So it has been stored for a number of at least 6 months. Then what we did was we crashed them and used acetone, acetic acid, and water. And what we did was with acetic, we used 79, 29, and 0.5% respectively. And what we did was we used rotary vapor to take out all the acetone in it, and then the solid we added some DMSO to it, and we introduced into the PDA media. I hope I answered the question though.
All right, thank you. And one last question is, this comes from Dr. Barry Tillman. Did you see a relationship between resistance and visible seed coat colors?
Well, based on that preliminary results we had, we've also requested new USDA lines based on just seed coat color. And because of the COVID, we are about to receive it, hopefully maybe this week. And we are going to see whether we can relate seed coat color to it, because what we found out was the resistance lines that we found were produced from a particular geographical area, which has been the center of diversity of peanuts, which is Argentina and Paraguay. And they had a particular seed coat color. And we are hoping to see whether we could exploit just the seed coat color as a criteria to determine resistance.
Okay. All right. This concludes the time we have for presentations. Thank you very much for your presentation today.